This is critical thinking in growth, development, and genetics. And here are the questions. So here's the first one, where you have to identify the false statement. Here you have to identify the true statement. And here is a clinical problem. Let's look at this first question, which asks you to identify the false statement. And let's look at the statements. So here it says, normally only one secondary oocyte is released into the peritoneal cavity. Now this is true. At ovulation, normally only one secondary oocyte is released, and it is released into the peritoneal cavity. Occasionally, only occasionally, you may have two released, and if two oocytes are released and they are fertilized by two different sperms, you get what are known as fraternal twins. And hence those twins need not necessarily be of the same sex, but this is only occasionally because each ovary ovulates alternately. Let's look at the next one, and which says both the chromosome number and the amount of DNA in a sperm or an ovum is half that of a cell elsewhere in the body. Now this again is true, and here notice I mentioned the word ovum, which in the females the secondary oocyte becomes an ovum only if it is fertilized, and so it is the ovum which has half the chromosome number and half the DNA, and the sperm of course also has half the chromosome number and half the DNA. And I'd like to show you an image. And here let us look at this picture where we're looking at gametogenesis. So it is spermatogenesis in the male and oogenesis in the female. So we begin with the spermatogonium or the oogonium, and that grows or enlarges and becomes the primary spermatocyte, which you see here. There's only enlargement, it just grows in size, and the counterpart of the primary spermatocyte is the primary oocyte. So this is like a regular cell which has 46 chromosomes, and at this point it has a normal cell, let's say, has 2N DNA as is shown here. When it becomes a primary spermatocyte or a primary oocyte, at that time the only change that occurs is that the DNA duplicates so that there can be shuffling of genes. So there is, let's say, it duplicates from 2N to 4N. So here also you can see in a primary oocyte I've shown you that it is 4N. Now in order to provide gametes, which are sperms and ova, you have to make the number half. The chromosome number from 46 has to become 23, and the DNA, originally it was 2N, that's that of a regular cell. So the DNA has to become N. Remember, 4N is not regular, it has duplicated. So this is what we are aiming for. So let's look at the process here. So the first meiotic division, from primary spermatocyte, it becomes what is known as a secondary spermatocyte. And from a primary oocyte, this is also a primary oocyte, it becomes a secondary oocyte. So at this point everything becomes half. So 46 chromosomes becomes 23 chromosomes, or 22 plus X and 22 plus Y in the case of the sperms, and 22 plus X in the case of the secondary oocyte, and the DNA from 4N becomes 2N. So this is what is seen in a secondary spermatocyte and in a secondary oocyte. So you have 23 chromosomes, of which one is a sex chromosome, and 2N DNA. Now, in the female, the secondary oocyte, only if it is fertilized will it continue. The process of oogenesis will continue. Otherwise, this secondary oocyte is this is the one that is liberated into the peritoneal cavity at ovulation. If it's not fertilized, it just passes out of the body. In the males, however, the process continues. So the secondary spermatocyte then forms spermatids. Now, in spermatids, the chromosome number remains the same, but the DNA now becomes half. So you can see that the DNA is half, which is what we aim to get. And then these spermatids undergo spermiogenesis, which is just a morphological change, and they become sperms. So sperms therefore also have 23 plus N. Now, should this secondary oocyte be fertilized, what will happen is that this will form an ovum, and in the ovum, so you can see it's called a mature oocyte and ovum, so you'll have 23 chromosomes and the DNA will become half. The chromosomes don't half at this time, it's only the DNA which becomes half, because this second meiotic division is very similar to mitosis. And then of course you have 3 polar bodies. So you can see that's how it happens. And I just want to show you another image which is really nice and should help you. So here you can see meiosis in the female. You begin with the oogonium, which enlarges and becomes a primary oocyte, and then you can see the DNA duplicate. So here, let's say there were 46 chromosomes and 2N DNA. Now you can see the threads are duplicating, so there's 46 chromosomes and 4N DNA. This then divides, so this is a primary oocyte, so it divides and becomes the secondary oocyte, which will have 23 chromosomes, and you can see 2N DNA, and it forms one polar body. Each one of them divides, and so you get 4 at the end. So then this is the ovum, which will have 23 chromosomes and N DNA. You can see the DNA has become half of this. There were 4 here, so you can see this half, and then you have the 3 polar bodies. So to come back to this statement, so this statement says both the chromosome number and the amount of DNA in a sperm and an ovum is half that of a cell elsewhere in the body. So this is also true. The third statement, which is HCG production, human chorionic gonadotropin production, which is produced by the placenta, increases in situations where the placenta is large. This is also true because HCG production increases where they are twins. If they are twins, obviously the placental size will be large. It increases in conditions which are known as hydatiform mole, which where the placenta sort of really enlarges and becomes like small vesicles. In diabetes also, because diabetic when the mother is diabetic, she tends to have large babies. So diabetic mothers, when the mother is diabetic, she tends to have large babies and the placenta is very large. On the other hand, you'll notice in ectopic pregnancy, the HCG production will be small because the placenta is very small, especially since ectopic pregnancies usually occur in the fallopian tube. So there's not enough space. Let's look at this fourth statement, which says gamete intrafallopian transfer, and this is called gift technique, is a method of in vitro fertilization. Now, in vitro fertilization means where there is fertilization outside the body. Now, in gamete intrafallopian transfer, read the words carefully. It says gametes are transferred into the fallopian tube. So you have the sperms and you have an ovum. Both are placed inside the fallopian tubes. And when the sperm fertilizes the ovum, the fertilization therefore occurs in the fallopian tube. So therefore, this is not a method of in vitro fertilization. So this is the false statement, and hence the answer to this question. But let's look at the last one, which says skin color is expressed through multiple alleles, and this is true. That's why you have the ranges of skin color. You know, when people of different skin colors marry, the children may be of varied color. The skin color may be varied, and that's because skin color is expressed through multiple alleles. Let's look at the next question, where you have to identify the true statement. And the first one is the process of spermatogenesis and oogenesis is exactly similar. Now this is false, and as I showed you in the previous in that image, and let's go back. And as I showed you here, notice that these 4 spermatids, they form 4 sperms, and this process goes on till the sperms are formed. Whereas this is one main difference. Whereas in oogenesis, notice the secondary secondary oocyte will form an ovum only if it is fertilized. So if it's not fertilized, the process is not completed. So that's another difference. So here, and the second difference is that there are 4 functional cells. Here we have only 1 functional cell, and we have 3 polar bodies which are non-functional because we want one cell to have all the cytoplasm, and also we don't want a population explosion. So these are some 2 major differences which occurs. And the other thing is that oogenesis begins before birth, whereas spermatogenesis does not. It begins at puberty. So you can see that there are quite a few differences, so they're not exactly similar. So here, this is false. This sperms attain complete motility in the female genital tract. This is true. Sperms attain a great degree of motility in the epididymis, but only when they are deposited in the female genital tract do they attain complete motility. So this is true. But let's look at the other one. Identical twins are formed when two sperms fertilize a secondary oocyte. This is false because identical twins are formed when one sperm fertilizes a secondary oocyte and then the zygote divides into two and each part then forms an individual. Fraternal twins are formed when two sperms fertilize two secondary oocytes. Now, in this case, when two sperms fertilize one secondary oocyte, you will have more than the normal number of chromosomes because you'll have 23 here for the sperm, another sperm 23, and that for the oocyte. So you get um, 69 chromosomes, which is, then life is not possible. So actually, you get a non functional zygote, and the zygote just dies. So this is false. The fourth statement, which is amniocentesis, is a procedure where you look at fetal urine. This is also false. And in amniocentesis, what you're looking at is the fetal cells, the cells which are from the skin, which get sort of abraded and are deposited in the amniotic fluid. And you're really looking at the fetal cells to look at chromosomal abnormalities. And number five, the son inherits X-linked diseases from the father. This is also false because the son is a male and he has XY chromosomes, the sex chromosomes. The Y comes from the father, the X comes from the mother. So therefore, X-linked diseases, the son always inherits from the mother, not from the father. So therefore, in this, the answer to this statement is this is the true. This one here is the true statement. Now, at the next question here, which is a clinical problem. So here it tells you that T for tall, the trait is T. The capital T is dominant over T, which is for short. Now it tells you that 50% of the offspring in any pregnancy will be short. It gives you that this is what is going to happen. So you need to find out what is the genotype of the parents. So now the way we can do this is, so let's make a Punnett square and let's work with the information that we have. So we see that 50% of the offspring in any pregnancy will be short. So short has to be recessive because we said T was tall, big T is tall, is dominant, and small little t is short. So if someone has to be short, their genotype has to be this way. So it tells you that 50% of the offsprings will be short. So let's just put 50% of the offspring will be this way. And so we just put it this way. So we know we have to work with this. Then it asks you what is the genotype of the parents. So let's work with this. So we know that let's look at this question, which is a clinical problem, which tells you about height, and it gives you a situation where T, which is uppercase T, is for tall and is a dominant gene over lowercase t, which is short. Then it tells you that 50% of the offspring in any pregnancy will be short. So they will be short. It tells you that. And then asks you what is the genotype of the parents. So here you're working backwards. You're given what the result will be and asked to find out what is the genotype of the parents. So let us look at, let me go back to red color and let's make a Punnett square and work with the information that we already have. So we know that the offspring are going to be short, which is they will be like this, and 50% are short. So out of 4 possible combinations, 2 will be this way. So I'm going to write the 2 this in this part here. We could write it anyway, so we can write it this way. Now, obviously, both parents cannot be homozygous short because if both parents were short, so that means the father and mother were like this, we would have all 100% would be short. So both parents can't be that. So we can erase this. So let's erase this. And the same thing also goes for both parents cannot be homozygous tall, because then all offsprings would be tall. Because if you had all capital T's, we wouldn't end up with a lowercase t at all. So let's work with, say, both parents are heterozygous. So they have one tall gene and one short gene. So they have one tall gene and one short gene. And let's see what the combinations will be. So this would be this way. You work, this would be this way. Now this would be this way, right? So then only 25% would be short. So this doesn't fit the, so, one parent being, or both parents being heterozygous also does not work. So we've discounted this one and we said they can't be this. And then both parents being heterozygous is also not, doesn't work out. So again, let me go back to, um, let's work out what the possibilities can be. And here. And so we come back to this. And okay, let me erase this one. Now let's say we have one parent who is tall, uh, and one parent, because that's what we are left with, and one parent who is homozygous and short. Now, then you will find that these are the possibilities you will get. This way, this way, and this will also be a capital T, and this will also. So all children will be tall. So even this does not work out. So therefore, this is also not, does not work out. So we can see that this is most likely the right answer. And let's work this and see if we can get that. So I'm going to erase this, erase this, and erase this. So we had that one parent is heterozygous tall and one parent is short. And let's see if we get 50% as short. So we get this way. This and this works out this way. This and this will be a capital T and a little t. So I can erase this. And this is little t. So we can see here we do have 50%. I just had it in the wrong place, but it's okay. We do have 50% who are going to be short and 50% who are going to be tall, but they are going to be heterozygous. So therefore the answer is this one. So this is how you're going to work out backwards when you need to find out what is the genotype.